So yeah, I mean, we can.
So you don't see with your eyes, right? We're not cameras. We're actually cognitive agents, and we're actually seeing with our brains. So this is a kind of classical cognitive neuroscience lab. So we're going to measure ERPs, which are event-related potential, which is a form of EEG, to understand how we understand the world, and visually, cognitively, how those different processes are implemented in the brain. And this lab is basically built to facilitate this whole data collection and data analysis process. What we would reveal is that vision is actually a very, I want to say, interactive process, whereby what you think, what you expect, what you need to do with the visual information actually constrains and basically leads you to particular interpretations. They're going to see different kinds of visual stimulation. This can be different objects, different scenes, different words, whatever it is that you're investigating. And then people would respond to those actual stimuli. Okay? Now, critically, while they're doing that, we're actually measuring their brain responses. How do we do that? They're going to sit with a cap. Okay? This cap is going to be connected with 64 electrodes, so 64 channels. That's a pretty good density kind of cap. Okay. And those electrodes are going to be connected to this amplifier. This amplifier, through this orange fiber optic, is going to send its information to our computers. This has multiple of applications. So in terms of education, you can think about looking, let's say, at dyslexic kids, right, and how their temporal processing of certain stimuli occurs. You can look at, attention deficits as well. You can also think about the more applied military applications. So for example, you have, a UAV operator, and you got to keep his vigilance, right? I mean, he's all those long hours and he's, staring at the screen. Let's say you have a measurement of his online attentional system, and then all of a sudden you see this drop in this EEG marker. Then you can send a feedback to that observer and just say, wake up, it's, you're kind of tapering off, right? Refocus, right. And again, this is a direct measurement, so you don't have to ask someone, did you think that this helped or not? You don't have to, they don't need to rate something. You're tapping directly into the brain. And that's kind of, really, it's fantastic.
Just a block, a single block. Now the participant is going to see 12 blocks, and so she's going to sit there for about